So we can't really afford to fall behind on the first day of class. So I'm going to do this chapter by recording it. And there's nothing in here that's so difficult that this isn't a good way to begin. However, I can't wait to meet all of you in class. So this is the book that you're using. You really can't use the 3rd edition because there were significant changes to the 4th edition, but you can certainly use an international edition. So about the book, I really do use the book in the course. It's critical. And part of that reason is because I like the questions they ask. On every section, at the beginning of every section, you'll find learning outcomes. Those are great for understanding what you're supposed to get out of that section. And then at the end of a couple of sections, there will be something called critical thinking boxes, and they're yellow. And those I use to pretty much come up with the essay questions on the exams. We also use them in class when we do the class participation activities. So they are an excellent way between learning outcomes and critical thinking to understand whether you're getting what you need to get out of the chapter. Okay. On every PowerPoint slide for every lecture, I will give you terms that you need to know and critical thinking questions from the ends of the chapters that I think are important. And then I'll also include any other little things that I think are worth knowing. That probably will show up on the exam if I take the time to do that. But again, you're going to learn a lot of material in this class. I can't test you on all of it, but you are responsible for it, and then I can test you on any of those things that I hold you responsible for. Whenever I'm not going to have you responsible for something in a chapter, I will let you know not to pay attention to something in a particular chapter. All right, so the first part of this is we have to go through the history of microbiology because it's important to understand where knowledge of microbes has led us. First of all, microbes exist in every environment on the earth. There's no environment where we have not found some kind of microbial life, usually a bacteria, sometimes a virus, but we found them in deep caves, radioactive caves, ice, glaciers, hot water vents, the atmosphere, rocks. There just is not anywhere that we have not found microbes. All right, on the slide that I showed you where you have to know the key players of microbiology, we have to understand why they started their investigations. And the first point to start from is what happened before the microscope was invented. Before the microscope was invented, all diseases and illnesses and fermenting of wine looked like something mysterious was going on. It was accounted for by religion, by curses, by bad luck, all those kinds of things. But the advent of the microscope, and particularly the grinding of lenses, was what turned it all around, that there was this unseen world. The first person to really grind high-quality lenses was Van Leeuwenhoek. He was able to grind lenses that allowed magnification up to 200 times, and he published a lot of his data into the national academies. However, when he died, he did not pass on how he ground his lenses, and it was lost for another almost 100 years before someone else could figure out how to grind his lenses so well. But in the meantime, he had taken samples of everything, things between his teeth, things outside, and he just looked at everything he could possibly find in the world and was the one who coined the term animalcules. Again, his knowledge was lost once he died with his lenses. Once we knew that these things were around, we needed a classification system so that people around the world could talk about them. And they came up with 6 areas. Some of this came later with the advent of Carl Woese. You'll find out who he is in the chapter. But there are 6 classifications, and you're going to know the main characteristics of each classification. Now I have a couple slides that go into that, and I think you should just pull out the most important points, and I've tried to make those in red. So if you go through bacteria and archaea, understand that while they are both prokaryotes, they do not have a nucleus, they do have differences. The most important difference is in their cell wall. Bacteria have a highly linked carbohydrate called peptidoglycan. That's down here, peptidoglycan. And archaea have a completely different cell wall, cell membrane structure. They just don't use that. All right, this is a nice little photo that shows you the difference in size between these things. This is a nucleus, and you can tell that bacteria are just fundamentally smaller. And I know most of you have used a microscope before, but if you haven't seen bacteria under a microscope, you're going to be very surprised. They're very, very tiny and hard to see. Even at 1000x magnification, these things are very small. There is no detail to be observed. All right, so we have fungi, which are eukaryotes. They have a nucleus. They have cell walls, but it's not cellulose, it's chitin. It's that hard stuff that you find on bugs when you squish them. It's called chitin. Fungi are composed of molds that you see growing on your bread or other places, even in blue cheese. And yeasts can be the ones that produce wine and beer and alcoholic beverages, or they can be the kind that are pathogenic and cause disease in humans. Here's some nice pictures. Of course, they won't look like this in our lab, but they're still, fungi in particular, are very beautiful under a microscope. Protozoa are eukaryotes. They're single-celled. They tend to live in water environments. The ones that live in animal hosts tend to be parasites. They can reproduce asexually or sexually, just like fungi. And their particular claim to fame is that they can move. They have locomotion, and they produce locomotion by pseudopods. They extend pieces of their cell membrane to pull themselves in a direction. They have cilia, like Paramecium has cilia, and they have a flagella. Now the flagella between a bacteria and a eukaryote are structured differently, but we'll find that out in a different chapter. Here's some photos. Algae, their claim to fame is photosynthesis. They are capable of using the sunlight to make energy, but they're not plants, they're algae. Here's a parasite. This is a nice worm infecting an animal because you see a red blood cell. This is a virus. So while we saw that many bacteria can fit inside of an animal cell, many viruses can fit inside even a bacterial cell. We can't see viruses with a light microscope. All right, so what were the 4 important questions we need to track through this chapter? And they are, what happened to spontaneous generation? It was finally disproved by Pasteur. How did he disprove it? Why did he want to disprove it? What was fermentation? Again, a big thing of Louis Pasteur, because water was not safe to drink. People knew that it made them sick, so they depended upon wine, which would either come out very well and be drinkable, or sometimes it would come out very acidic like vinegar and they couldn't drink it, and they didn't know why. So that's why they wanted to know about fermentation. The philosophers of the period, which were what scientists were called at that time, also wanted to know what caused disease. They observed that people weren't necessarily sick all the time, and that there would be epidemics in one place and not in another place, that diseases shared certain characteristics and symptoms and outcomes, whether deadly or not. And then they wanted to know, how can we prevent these kinds of epidemics? People were, starting to live in large cities and getting very sick and having nasty epidemics. So basically, they observed from animal life to the things they could see that were very small without a microscope. They knew that things could produce asexually, like plants, sexually, like animals, and then the rest, the very tiny things, they thought that they arose from non-living matter, that you could put a piece of meat in a jar and maggots would arise spontaneously from this. And a bunch of scientists performed some experiments to try and refute that, because they just didn't believe it. They didn't think that would happen. So the first one was Francisco Redi. He took a chunk of meat and he put it in 3 different jars. One was completely open, one closed, and one had just a bit of gauze over it. And sure enough, the one that was completely closed and the gauze didn't grow maggots. Well, we know it's because the flies didn't land on it. But what they said was Francisco Redi's experiment was flawed because he kept out the vital force. Now, don't forget the time period that we're talking about here, and they felt that everything needed a vital force, and he left that out, so therefore they didn't believe his experiment. Next up came this fellow named Spallanzani, and he said that another person named Needham, who also tried an experiment with sealed vials, didn't allow any air in, so how could anything have survived anyway? So he did an experiment where he definitely boiled it and again sealed tubes, and that they didn't believe that one either because, again, there was no air. So who fixed it? Louis Pasteur took all of those arguments into account, allowing in the vital force, allowing in air for something that's living to survive. And what he came up with is this great thing with flasks. And you'll have to go to your book. Somehow I lost my slide on that. But he used a gooseneck flask. And because the gooseneck allowed the vital force, it allowed oxygen in, no one could refute Louis Pasteur's experiment. So if you look on your textbook on page 10, it shows these really lovely gooseneck flasks. And that was the experiment that led to the demise of spontaneous generation. It took over 100 years to get rid of that, from the start of early experiments to the end. So basically, they also believed Pasteur because he did a finely tuned process of the scientific method. And that is also on page 10 if you need a review of that. There will be occasions where you're asked to evaluate an experiment, and you should understand the steps of what constitutes a good science experiment. All right, fermentation. Louis Pasteur got involved in this. There was big money in this. Fermentation was very valuable to people who had vineyards, and they wanted to know why sometimes they had great wine and other times they had great vinegar. So Louis Pasteur investigated what kind of organisms he found in the wine. There's those gooseneck flasks. It's out of order. So what happened is he took grape juice and he basically lightly boiled it. He thought he could kill some of those things that Van Leeuwenhoek saw with his microscopes and other people were seeing with microscopes. And he thought that he would just boil it a little bit. And then he introduced yeast into it to see what would happen. Well, he found that if he let yeast and bacteria into the flask, he would have vinegar. If he excluded the bacteria by lightly pasteurizing it, giving it some heat for a short amount of time, then only the yeast would grow and he would have fermented beverage. It would turn into wine. So he's basically the person that discovered that bacteria were bad in wine and yeast were good, and yeast are eukaryotes. It also led to the process of pasteurizing many other liquids, as you know. And now there are different types of pasteurization that you're going to learn about in other chapters. So he started the field of industrial microbiology. So you're going to be expected to be familiar with some of the early experiments that led to the demise of spontaneous generation. And you should definitely know about Pasteur. He's famous for many other things in bacteriology. Here are just some industrial uses of microbes. Very interesting. I might ask you some of these things. I might say, what are 2 or 3 things that microbes help produce? Fermentation was demonstrated by a man named Buchner, who showed that you didn't need a living cell to ferment something. He discovered that enzymes could perform the chemical reactions that resulted in fermentation. And you're going to learn all about that when we study the biochemistry in one of our chapters of microbes. Okay, now that we know there are microbes, now that we could see them, now that we knew they didn't rise by spontaneous generation, along comes a fellow named Koch, C-O-C-H, who wanted to know, well, what causes disease then? Are these possible animalcules implicated in disease? So he found a way to grow these in a laboratory. If you can't grow something in a laboratory, you can't study it. So he took the combination of some staining techniques that had been discovered, what people had drawn, some micrographs. He was able to develop media, so this is just a nasty mix of things that could be boiled and turned into a food for microbes. He used petri dishes and gelatin, and he grew bacteria, just like you're going to in a lab. He laid down 4 postulates. He said that an agent, which I'm going to use that word a lot, so just get used to it, an agent of disease must be found in every case of the disease and must not be found in healthy hosts. Makes sense. He said it must be able to be isolated and grown in some of his petri dishes and then reintroduced into a healthy host. The healthy host would have to get sick from that introduced agent, and then he would have to be able to re-isolate it from that sick animal, again on his petri dishes. If he could do that from sick to a dish, back to a healthy animal, back to a dish, and it was the same agent, the same bacteria, the same fungi, then that was what caused disease. And he was able to do that for a number of diseases. Now here's a nice table of other scientists who pursued disease and what caused them for various reasons. You've probably heard, you haven't heard of brucellosis probably, but all of the other ones you've probably heard of. And these are the bacteria that you'll start learning the names of. You'll even have to know how to spell them. And we will study many of these during the semester. All right. When Koch started discovering these bacteria, people naturally wanted to see if they could find out characteristics about them using a microscope. Bacteria are clear. They need to be stained to see them. The most promising technique, and the one that you will use in the laboratory almost every single lab session, is called a Gram stain. And what the Gram stain does is differentiate between a Gram-negative bacteria and a Gram-positive bacteria. All that means is that Gram-negative bacteria have a very thin layer of peptidoglycan. Gram-positive bacteria have a very thick layer of peptidoglycan. In another chapter, you will find out the structure of peptidoglycan. You will find out other characteristics of Gram-negative bacteria and Gram-positive bacteria. But for now, Gram-negative are red, like this one, and E. coli is the best example of a Gram-negative bacteria. Gram-positive bacteria come out purple due to the Gram stain. And a good example of those would be a Staphylococcus. Okay. Then, once we knew what caused bacteria, what caused diseases, bacteria, we wanted to know how to prevent them. So if we know what causes disease, can we prevent people getting it? And the key players in this epic of microbiology were Semmelweis, Joseph Lister, Florence Nightingale, John Snow, Edward Jenner, and Ehrlich. I forget his first name. So Semmelweis actually worked in hospitals, helping to deliver babies and perform autopsies, and he found out that hand washing prevented death of women after childbirth. And I want you to look him up and read about him. It's fascinating. He almost halted childbirth death in its tracks. And I want you to read about what happened with his colleagues. It's a very sad story. That's in your book. Joseph Lister discovered chemicals that would kill microbes, and he said that when he washed his hands with these carbolic acids, that his patients lived longer after surgery, because in those days surgery was almost a death sentence because of infection. Florence Nightingale brought clean techniques to the hospital environment and nursing. John Snow was the father of epidemiology, and he's another person you have to look up, use the book. I encourage you to use the internet because it's fascinating the way he used maps to plot out where the heart of a disease was occurring, when it started to wane, where the index case might have occurred. The index case is the first person who contracts a disease. Just recently, they were able to track down the index case for the Ebola epidemic. Right now, they're looking for the index case for the measles epidemic that's going on that started at Disneyland. We actually use most of the same techniques John Snow invented, so you have to read about him. Ask me questions in class about Semmelweis and Snow. I want to discuss it. Jenner is responsible for developing vaccines, vaccinations, variolation. He's the father of immunology. I want to hear what you have to say about Edward Jenner and his smallpox and cowpox vaccinations. Ehrlich was the one who is considered the father of chemotherapy, or the father of developing drugs to treat diseases. Chemotherapy is nothing but drugs to treat disease. If you take an antibiotic, technically you're undergoing chemotherapy, even though by custom we generally use that to describe drugs that treat cancer. So Ehrlich came up with a drug to treat syphilis, actually. This is a nice graph that you can look at, and it tells the people and what they did in the field of microbiology. There's all kinds of fields that involve microbiology. All right, we've got what falls under this umbrella is bacteriology. Studying about algae is phycology, mycology. I'm not going to ask you these, but you should know that if I say virology, I'm talking about viruses, parasitology. You can study metabolism by microbes is actually fascinating. And of course, genetics is huge in microbes. They taught us so much of what we know about genetics was started in microbes. Here's some other fields that are involved with microbiology. One of the most interesting is foods, remediation, biotechnology, even agriculture. People tend to give agriculture short shrift, particularly here, But in the Midwest, it's huge. Microbiology and agriculture are huge. All right. The biochemistry that has to do with microbes basically started with Pasteur, went through these two guys that are in your book because they did work with enzymes and test tubes and things. They did the biochemistry. And then they applied it to, again, the field of agriculture, disease, and treatment. Then, all of this, from not being able to see anything because of no microscopes, led all the way up to genes. Genetics was first studied in microbes, particularly in pneumonia because it was killing people in the right up until the 40s. So basically what we know about microbial genetics obviously came from microbes. What we know about even animal genetics was started in microbes. The field of molecular biology came out of microbiology. Recombinant DNA technology would not exist without microbes, and gene therapy is being carried out with the aid of microbes and viruses. All right. If you have not taken BI 253, you should go back and review these scientists because they made giant leaps forward, and I will assume that you will know the basics of what they did. Avery, McLeod, and McCarty, Beadle and Tatum. I might even mention the blender experiments with Hershey and Chase. You should certainly review how genetic information is replicated, transcribed, and translated. We will be doing a genetics chapter, and if you don't have the fundamentals down, you will be lost in that chapter. So I beg you to look through chapter 2 of the textbook. You'll notice that it is not on our syllabus. It's expected that you review that yourself. All right. We're going to talk a little bit about recombinant DNA in using microbes. You're actually going to transform a bacteria and make it glow this semester. You're going to see some of the drugs that we've been able to engineer thanks to microbiology. We're going to talk a little bit about gene therapy, not a lot. We're going to talk a little bit how microbes are involved in cycling major nutrients in the environment. And of course, I wish we could do more on marine microbiology, but we can't. And then we're going to talk a little bit about serology, immunology, and we're going to cover antibiotics and how they work to kill microbes. Fungus, very interesting. Of course, that's what Fleming discovered. He discovered that penicillin would inhibit the growth of bacteria, and you're going to do an experiment almost identical to this. You're also going to use other things like household products to see if it will inhibit the growth of bacteria. Here are the critical thinking questions of the chapter. See if you can answer them by reading the chapter. I want to discuss them in class. We're starting on a Wednesday, so that's the day that class participation consists of critical thinking questions. So how this works is you're going to come in, and at some point during the class, I'm going to have you write down answers to these critical thinking questions. Your participation is based on how we can discuss that as a class. I may even have you write them down and turn them in once in a while, so you should be prepared. You only have a few minutes. So if you review the critical thinking questions before you get to class, you'll be prepared to answer them in the short time allowed for the activity. And I certainly would love to discuss them once we get to class. So read these over, jot down some answers, and we'll see how far you get. It'll help you practice for the exam. This semester, you will be asked to read one book. You can choose The Hot Zone, Demon in the Freezer, Wisdom of the Whores, or The Wildlife of Our Bodies. The Wildlife of Our Bodies is about parasites, and it's fascinating the impact that parasites have had on disease and our bodies. You will be very surprised. It's a very easy read. The Wisdom of the Whores, Bureaucrats, Brothels, and the Business of AIDS is fascinating if you would like to know the numbers behind AIDS. It's very educational. It's sometimes unsettling, but you know, we haven't been able to cure AIDS or come up with a vaccine since the 80s. So if you're interested in some of the statistics behind it and why we haven't been able to come up with some answers to AIDS, you read that one. She's very witty, and she was an AIDS researcher. If you like you know, international intrigue and where storage of some of our more dangerous microbes are kept and what's happened to them, then you should read Demon in the Freezer. It is a factual account of the storage of microbes that are potential bioweapons. The hot zone is particularly relevant because this was written before Ebola came on the international scene a few months ago. And of course, that epidemic is not yet done. It's just that the numbers right now are not increasing. It seems to be under control. But this was really quite prophetic that Preston covered this and wrote the book, The Hot Zone. All right. So basically, we'll talk about this in class. You can come up with your questions, and I have a typo in there. But the first 2 chapters, prove to me that you've read the first 2 chapters by the first exam. You have to do the next 3 chapters by the second exam, and you have to have the project finished a week after the third exam. Now stop rolling your eyes. The project is up to you. You can do a blog entries for each chapter. I had a student last semester do this beautiful thing with books that you pull off a shelf digitally, and she wrote her personal responses to events in the chapters. You can do the blog however you want. You can be very creative about it, do it as a news story. We had other people do music videos. That was really entertaining. We had another student do a pro versus con on Ebola, and he portrayed himself as a doctor in one scene, and someone arguing about it in another scene. I don't know how he did that. It was wonderful. Radio broadcasts, just the sky's the limit. Be as creative as you want to be. I had one girl, if you say that to yourself, oh, I'm not that creative. I had one student who just recorded herself talking about the book, and she didn't think it would be very good. And yet, it was really great to see what she thought of the book and her insight into what was going on, even as she questioned, well, is that what it really meant? This is what it meant to me. And that's completely valid. So get creative, get the books. They're very inexpensive, especially for the downloads, or maybe someone from last semester has the book, and start reading now. And I'd love to discuss them in class. That's the end of my PowerPoint, and I will see you tomorrow afternoon as long as class is in session. You can reach me by email, and see you soon. Bye.